Imagine being able to guide immune cells as if they were tiny soldiers, steering them through microscopic mazes, exposing them to precise chemical cues, or watching them engage with pathogens one on one, all in a controlled, engineered environment no bigger than a coin. This is the promise of microfluidics, the science of manipulating fluids at the microscale to mimic and probe the complex environments of the human body. When applied to immunology, microfluidics offers a revolutionary platform for studying, regulating, and even programming the behavior of immune cells with unprecedented precision. At first glance, microfluidic chips might look like miniature circuit boards, but instead of wiring electricity, they channel droplets, cells, and molecules. These lab-on-a-chip devices can replicate aspects of the immune microenvironment, from lymph nodes and inflamed tissues to tumor borders, allowing scientists to isolate specific interactions and decipher the language immune cells use to detect threats, activate responses, or coordinate with each other. So, how can microfluidics regulate immune cell behavior, not just study it? The answer lies in the fine-tuned control over biochemical gradients, mechanical cues, and cell-to-cell communication that microfluidics enables. In the body, immune cells migrate in response to chemical signals called chemokines, experience physical forces in tissues, and make decisions based on the local molecular milieu. Microfluidic devices can replicate these conditions in vitro, allowing researchers to tweak individual parameters and observe how immune cells respond. Consider T cells navigating a complex tissue. A microfluidic chip can mimic this environment by creating a chemokine gradient across narrow channels, directing T cell migration in real time. By adjusting the steepness of this gradient or introducing competing signals, researchers can uncover how T cells prioritize targets, a key question for improving immunotherapies such as CAR-T cells used in cancer treatment. These chips can also mimic the immune synapse, the highly specialized contact point between a T cell and an antigen-presenting cell, enabling nanoscale analysis of activation signals and cytotoxic responses. What makes microfluidics especially powerful is its capacity for single-cell analysis. Unlike bulk assays that average data across thousands of cells, microfluidic platforms can isolate and monitor individual immune cells, revealing variability in activation thresholds, cytokine production, or memory formation. This is vital in understanding heterogeneity in immune responses, such as why some patients respond better to vaccines or immunotherapies than others. Microfluidic systems also allow for mechanical manipulation. Immune cells are sensitive to physical forces, pressure, stiffness, confinement. Using deformable microchannels, researchers can study how cells like macrophages or neutrophils change shape to squeeze through tissues, or how stiffness affects dendritic cell maturation. These findings are critical for designing biomaterials and implants that interact favorably with the immune system. Another cutting-edge application is in the co-culture of multiple cell types. Microfluidic chips can house immune cells alongside cancer cells, endothelial cells, or pathogens in distinct but communicating compartments. This setup enables the modeling of tumor-immune interactions, immune evasion strategies, or the testing of immune modulatory drugs in real time. For example, researchers can observe how checkpoint inhibitors unblock T-cell attacks on cancer cells within a tumor-mimicking microenvironment. Which materials and designs make this possible? Most microfluidic devices are fabricated from PDMS, polydimethylsiloxane, a biocompatible silicone that allows for optical imaging and oxygen permeability. Channel designs can range from simple linear paths to intricate mazes mimicking capillary networks or lymphatic flows. Integration with optical sensors, impedance measurements, or fluorescent reporters enables dynamic monitoring of cellular functions. Looking ahead, the integration of AI, automation, and organ-on-chip systems is set to transform this field. AI can analyze vast imaging data from microfluidic experiments to identify behavioral patterns or predict immune responses. Fully automated chips may perform iterative experiments, adjusting stimuli based on previous outcomes, like a learning immune system in silico. Meanwhile, merging immune chips with good-on-chip or lung-on-chip systems opens doors to studying systemic immune regulation across barriers and tissues. Ultimately, microfluidics is not just a tool for observing immune behavior. It is a platform for manipulating and directing it, from fine-tuning vaccine formulations to testing personalized immunotherapies. It offers a new paradigm where the immune system is not only studied, but engineered, cell by cell, signal by signal. Could the future of precision immunology lie in the palm-sized architecture of a microfluidic chip? The mounting evidence suggests it just might. Microfluidics is the science of manipulating fluids at the submillimeter scale to create highly controlled environments for biological experimentation, allowing precise control over spatial and temporal parameters such as fluid flow, chemical gradients, and mechanical forces, which makes it a powerful tool for studying and influencing immune cell migration, activation, and signaling in real time. For example, microfluidic platforms can replicate interstitial fluid flow and gradient cues that guide T-cell movement during inflammation. And it's like designing a miniature obstacle course where each curve and slope influences how an athlete decides to move and react. Immune cells rely on chemotaxis, their ability to migrate in response to chemical gradients to locate infection or injury sites. And microfluidic devices can generate stable, reproducible gradients of cytokines, chemokines, and antigens to precisely guide cell movement and study migratory behavior, overcoming the variability of traditional assays while allowing dynamic modulation of gradient strength and direction. For example, a microfluidic chip generating an interleukin 8 I8 gradient showed how neutrophils adapt their directionality in response to changing concentrations, and it's like laying out a scent trail in a maze to watch how a trained dog sniffs out the strongest path to the target. Microfluidic platforms can sort immune cells based on physical properties, size, deformability, or molecular markers using integrated sensors and selective channels, enabling high-throughput phenotyping and real-time observation of subpopulations such as effector, memory, or regulatory T cells, while offering label-free, non-destructive alternatives to flow cytometry. For example, deterministic lateral displacement chips separate activated T cells from naive ones based on subtle changes in deformability and surface expression, and it's like filtering coins through a coin sorting machine, each slot designed to catch subtle differences in diameter or weight. By controlling microenvironmental cues such as shear stress, ligand presentation, and antigen exposure, microfluidics can modulate immune activation and tolerance, crucial for studying autoimmune reactions, vaccine responses, and tumor immune evasion. By replicating immunological synapses or presenting antigens in a pulsed manner to mimic physiological exposure. For example, a microfluidic platform presenting tumor antigens at varying densities revealed how dendritic cells fine-tune T-cell activation thresholds, and it's like adjusting the brightness of a signal lamp. Too dim and no one notices, too bright and everyone overreacts. Microfluidics enables the isolation and monitoring of individual immune cells, allowing researchers to dissect heterogeneity in immune responses at the single cell level. Essential because even genetically identical cells can behave differently under the same stimulus, and supports high-resolution temporal analysis of gene expression, cytokine secretion, and signaling dynamics. For example, a droplet-based microfluidic system captured single B cells and measured their antibody production profiles under stimulation. And it's like interviewing each soldier in a battalion to understand who's calm, who's aggressive, and who's about to retreat. Microfluidic culture systems model tumor immune interactions by housing cancer cells and immune cells in separate but interacting chambers, enabling study of immune infiltration, suppression, or activation in confined microenvironments, with spatial compartmentalization for visualization of immune synapse formation, checkpoint inhibition, or cytokine diffusion. For example, a tumor on chip device demonstrated that blocking PD1/PDL1 restored T cell killing ability in confined tumor microenvironments, and it's like observing a battle through a transparent wall, seeing how defenders and invaders strategize in real time. Microfluidics offers platforms for controlled delivery of vaccine formulations and for analyzing how antigen presenting cells prime T cells by using synthetic antigen presenting surfaces, droplet encapsulation, and pulse delivery to more accurately mimic in vivo conditions than bulk assays, thereby optimizing dose, timing, and delivery format for more effective immunization strategies. For example, liposome encapsulated mRNA vaccines tested on chip-based dendritic cell arrays revealed optimal timings for co-stimulation signal delivery. And it's like training soldiers with varying instructions and watching which methods produce the most vigilant and responsive recruits. Patient immune and cancer cells can be analyzed in microfluidic chips to evaluate responses to checkpoint inhibitors, cytokines, or engineered cell therapies, making microfluidics a powerful tool in personalized immunotherapy screening for rare or heterogeneous cancers. While small volume requirements reduce cost and allow multiplex testing, for example, a personalized microfluidic assay identified the most responsive CAR cell formulations for individual leukemia patients. And it's like test driving custom built engines on a scaled racetrack to find the best match for each vehicle. The next generation of microfluidic systems integrates machine learning, real-time imaging, and organ-on-chip technologies to simulate entire immune responses in complex tissues, with AI analyzing vast datasets generated by microfluidic assays to predict immune outcomes or optimize experimental design, paving the way for autonomous immunoengineering systems that design, test, and refine therapeutic strategies. For example, a lung-on-chip model with AI-guided analysis predicted cytokine storms in response to viral antigens before clinical symptoms emerged. And it's like building a miniature battlefield with automated drones that not only watch, but also suggest new defense strategies.